A very good morning, Professor Garg and my dear colleagues. My name is Kajol, and today I'll be giving a presentation of my seminar second. The the topic of my article is Solecidin containing Solanum torvon fruit extract prevents chronic constriction injury induced neuropathic pain in rats in silico and in vivo evidence of TRP1 receptor and cytokinin inhibitions. So Professor, this is the first page of our article, and it was published in 2023 in Molecular Neurobiology, which is the Springer journal. uh, Here is some minute details about the journal. The impact score of the journal is 5.12. The H index is 130. It was published in 2023. The type of the article is research article, and the name of the journal is Molecular Neurobiology. The publisher is Human Press, and the country is United States, and it is a Cuban journal without any publication fees. So let's talk about the graphical abstract. The graphical abstract was not published, but I made it so that you can easily understand the paper. So here's the graphical abstract. First, we prepare the ethanolic extract of Solanum torbum. The fruit was used to make the extract, and then it was used for the treatment of chronic constriction injury. In vivo, we check the behavioral, biochemical, and histopathological scale. The behavioral studies was conducted on days 0, 7, 14, and 21, and then we check the mechanical, thermal, and cold allodynia in Vistar rats. And the biochemical we estimated interleukin 6, TNF alpha, T bars, MPO level, SOT level, and reduced glutathione. The the in silico studies was also conducted of the article. Sorry for the uh, like, I think it's the second hour. Okay, so in silico studies, in silico we conducted the stimulation studies of solecidine with the specific binding with the TRP1 receptor, interleukin 6, and TNF alpha, which are the main receptor of the neuropathic pain. The binding was found to be minus 11.2 and 6.04, which is a very good binding with the specific receptor. So this was the this was the written abstract which I summarized below. So let's talk about the introduction. The neuropathic pain is a subtype of pain caused by the various sources such as lesion, injuries, or the accidents, which are the main reason for the nerve injury. And there are certain diseases such as neurological disorder, brain stroke. They are also causing the nerve injury, and which in turn causes to neuropathic pain, for which still we have no treatment. So the prevalence of this condition is very high, and the treatment which we have, they possess very higher adverse effects. And the common adverse effect of the current therapy is tolerance, addiction, and sedation, which causes a big issues for the patient. And uh, and why we have chosen this plant? Because this plant has given some very good antioxidant properties in previous researches. So let's talk about the material and methods. So we have taken the Vistar rats of either sex. Why of both sex? Because this injury is common in both male and female, and the Vistar rats, they are majorly used in research. And the collection of this plant, the plant was collected from the Kantholor Luduki district of Kerala, and the plant was identified by the Professor Jeswal. Uh, and how we prepared the extract: the fruit was macerated with 100% ethanol for 72 hours, resulting in extraction of 1500 gram of coarse powder. The extract was filtered, vacuum concentrated, and the syrup substance from that process was lyophilized to create the powder. It was kept for the upcoming trial at below 25 degree. So basically first we do the maceration with ethanol, and the powder was dried and lyophilized and stored at 25 degrees Celsius. The phytochemical structure standardization was done with TLC, thin layer chromatography, and HPTLC, the high performance thin layer chromatography. So these are the in vitro, like the in vitro antioxidant assay of the extract. First we did the DPPH assay, and this is the formula for estimation of DPPH assay of the extract. Then we check the ABTS activity of the extract following by this formula. The percentage inhibition and percentage scavenging, it gives the in vitro antioxidant activity of the extract. And later on the FRAP, which is also called as ferric reducing antioxidant potassium assay, was also estimated. The CCI, how it was induced? It was induced by the surgery. The sciatic nerve of the Wistar rats was ligated with some sutures, and then we perform the behavioral tests such as acetone test, pinpricks test, walking test, hot plate test, and then we further check the protein content, cytokinin level, and myeloperoxidase activity. And then further we estimated the histopathological evaluation of the sciatic nerve. And initially we did the induced fit docking stimulation studies, whether the psilocybin binds to the receptors or not. So these were the animals groups which were used in this activity. Basically 10 groups were used. 3 groups were used basically to check the mechanism of action of the standardized extract, and the 8th group which we checked. The first group is the normal control group, in which only we give the food and water. No surgery was done in this one. The second group was CCI group. In this we only do the surgery and nothing else. We don't give any treatment. The third group is sham control group. In this what we do basically, we do the some we expose the area, but we do not do the ligation. The another study is the in which we used extract plus pregabalin, which is the standardized drug for the neuropathic pain treatment, and the another drug is EESTF in CCI. The next drug is pregabalin, and the next one is psilocybin, which was procured from the Sigma Aldrich with the help of capsine, psilocybin, and the extract at 300 mg. We checked the possible mechanism of the extract, and these were the results of the T bars, which is antioxidant parameter. Then this is the results of GSH, SOT, CAT, or MPO, which is the MPO is the basically the biomarker for inflammation, and the CAT, SOT, reduced T bars. These are the antioxidant and anti-inflammatory parameters in the sciatic nerve of the rat. So let's talk about the results now. So first graph, it shows the HPLC graph of the pure psilocybin. Here you can show the peak at 545, and this one is the peak of extract psilocybin. And this shows the cold aerodynia, in which how we do that, we basically spray the acetone on the feet of the Wistar rats, so it will cause the pain. So you can check that in CCI this the value is very high. And this one is the hot plate. We basically place the animal on a hot plate and turn the hot plate on. It will cause again the pain. So here we check the again the criteria. Then with the pinprick test, with this also we causes the pain. And the walking test, we basically stain the leg of the rats, and then they walk, and then we check the pattern of the walking of them. Then we talk about the interleukin 6, 10, and TNF alpha in the Wistar rats, and then we check the histopathology in the specific groups. In normal control group, the first one is the normal, and another one is sham, and another one is CCI treated, and another one is pregabalin, which one is the D and the standard one, and the E and F is the uh, uh, and the another group is the extract at 100, and the another one is the extract at 300 mg. So these are the results of the acetone and other things. So here comes the docking result. Basically we can see that the prega the like the binding of solicitin on the TRP1 receptor. Here you can see that it binds specifically with the OH group. The Here we can see that the binding of the solicitin to the respective receptors. The first one is TRP1 receptor, and this one is the TNF alpha, and then this one is interleukin 6 receptor. So here we can see that the drug is actually binding to the specific receptors. So thank you so much, Professor Gar and my dearest classmate, and special thanks to Rahman for helping me at the last moment. Thank you so much.